So now we want to talk about bulk transport across the membrane. So far, we've looked at this idea of pores and channels. We're moving small molecules across the membrane. There's a protein that's either changing shape or has a hydrophilic hole through it to allow things to move through the membrane. But if we want to move something big across the membrane, like a whole protein complex or a large protein or an antibody, for example, or if we want to engulf food on the outside, like an amoeba is going to eat paramecium or a bacterium or something like that, we need a different mechanism. And that mechanism is going to be bulk transport. So bulk transport is still active transport. It takes energy. It's going to take a lot of manipulation of the membrane. And so let's take a look at exocytosis as our first example. So this is going to be bulk transport of stuff leaving the cell. So remember when we talked about the endomembrane system, we talked about proteins being processed in the ER, getting some carbohydrates added on there, some polish work being done in the Golgi apparatus, and then one destination was a secretory vesicle from the Golgi to release the contents that were on the inside of the vesicle to the outside of the cell. So again, this brings up the idea of topology, that when we set these things up as they're moving through this endomembrane system, that what is on one side of the membrane has to have a very specific destination. So when I'm on the inside of a secretory vesicle, it means I'm going to be on the outside of the cell when that vesicle fuses with the plasma membrane and is secreted. Exocytosis. So in all the pictures and the movies that you're going to see, this occurs very simplistically. You just see these fluid-like bubbles blend in with the membrane, and they meld and they fuse, and that's all great. But in fact, this is an oversimplification. Not something we're going to delve into in this course, but know that, surprise, surprise, it's more complicated than that. There's a whole team of proteins that are involved in bringing this vesicle to the plasma membrane and fusing those membranes in a very controlled manner. But when I do so, again, the inside of that vesicle becomes the outside of the cell, and those proteins, or those larger molecules, never cross a membrane and are now outside the cell. So exocytosis is exiting the cell. Endocytosis is going to be the opposite. And that's going to be bringing things from the outside of the cell into the cell. And there are three kind of categories on a basic level of endocytosis. So it's going to be phagocytosis, which is cellular eating, pinocytosis, cellular drinking, and receptor-mediated endocytosis. Let's look at each in turn. So if we look at phagocytosis, we're going to engulf a particle from outside the cell. Usually this is going to be a food vacuole that's going to form, and it's going to fuse with the lysosome to digest the contents to be broken down so the cell can use it. So when we look at that in this picture, we can see it's kind of non-discriminate. We're going to engulf whatever's to the outside of the cell, and the cytoplasm is actually going to form pseudopods in many cases, and reach out into the environment, form this food vacuole. And you can see in this transmission electron micrograph of a amoeba trying to engulf a much larger, a very large bacterium. So you see the food vacuole forming around it, attempting to form around this, and then ultimately this amoeba will digest this bacterium. So phagocytosis, eating, cellular eating. Pinocytosis is going to be much, much on a smaller scale, tiny gulping of the surrounding media. So these vesicles are much smaller. You can see, this is a great electron micrograph, you can see the tiny invaginations of the membrane that are going to pinch off to make small vesicles, kind of sampling the external media. And whatever happens to be there, the ions, the small soluble components, are going to be drawn into that vesicle. Pinocytosis. Now, receptor-mediated endocytosis is the last class of bulk transport that we're going to talk about, and this is very specific. So we're going to talk about receptors more later, but this idea that I can have a membrane protein that is exposed to the exterior of the cell and is looking for or has an affinity for a specific ligand. So a ligand is the general term for that which binds to a receptor. And so in this case, you can think about like the receptors that are lining your cells that are waiting for a hormone, a chemical signal from another part of your body that then they bind to and then send a message to the interior of your cell. And we're going to talk about that more later, but in this case, when we talk about receptor-mediated endocytosis, those receptors are going to send a very specific message. And that message is going to be to draw in the membrane and bring in these receptors and their respective ligands. So there's a very specific protein associated with this. It's called clathrin, which I encourage you to look up and read about to learn about this a little bit more deeply. But receptor-mediated endocytosis, the key difference here is that it's very, very specific. It does not occur until the receptor is triggered by binding to its ligand.